afternoon, everyone. Bit of a pivot now from drawing technology into ovarian cancer. Firstly, Janie, so proud of you. Congratulations. As Abby mentioned, I've been fortunate enough to be on the receiving end of Janie's leadership and guidance in my own career, which I'm very grateful for. So congrats, very proud of you. All right, I might begin my speech today by asking everyone to imagine something. So for the women in the room, imagine you're in a doctor's waiting room. And for the men, imagine some female significant other in your life, be it your wife, daughter, sister, mother, whoever it might be, also in the waiting room. You've just had an ultrasound and the doctor has found an ovarian mass. So you ask the obvious question, is it cancer? Today, far too often, the answer is, I don't know. Now, that's not because doctors aren't highly skilled, and it's not necessarily because the technology doesn't exist. It's simply because there's no reliable diagnostic test to differentiate a benign from a malignant mass. As a result, millions of women undergo surgery every single year simply to get a diagnosis. And it's not minor surgery either. In fact, it's really major. So we're talking removal of fallopian tubes, ovaries, leading to essentially instant infertility, menopause, and a host of other comorbidities that come along with that surgery. What makes this even worse is that most of these surgical cases, in fact up to 90%, are benign. So for me, that is probably one of the most extraordinary gaps left in modern medicine today. And that's the problem that Cleo is here to solve. How? So Cleo's developed a simple blood test that can accurately differentiate benign from malignant earlier and before surgery when the treatment matters the most. So how does that technology work? Our test is based on biomarkers. So biomarkers are naturally occurring proteins in your blood that give you important clues to what's happening in your body. So rather than relying on a single biomarker, we've got 8. So a panel of 8 biomarkers, including our patented biomarker, CXCL10. And we combine these with a proprietary algorithm that essentially give you a risk of malignancy algorithm. The remaining biomarkers are pretty well recognised and known in the ovarian cancer pathway. I'll point out one called CA125. It's one of the most widely used blood tests today as a standard of care, and it plays an important role in navigating ovarian cancer. But it has very well recognised limitations. CA125, it's not specific to ovarian cancer, so that means it's actually elevated in a number of benign conditions. So think endometriosis, pregnancy, uterine fibroids, and other inflammatory diseases. It can even be elevated when a woman does exercise, right? And what makes this even worse is that it's also women with ovarian cancer can also have normal CA125 levels. So as a result, it's highly inaccurate. So rather than replacing these biomarkers with other ones, we've incorporated them into our panel. So CXCL10, I like to think of as our secret ingredient. So CXCL10 is present early and in high levels in ovarian cancer development, making it a really powerful indicator. And so one of the things about Cleo's technology that I find probably the most compelling is that it's not just the science itself, because the science is honestly incredible. I did a lot of DD before I joined this company. It's that we developed the technology to fit within how healthcare already works and exists today. So doctors already order blood tests, pathology labs already run immunoassays every single day. So we're not asking clinicians to change the way they practice medicine. We're simply equipping them with better tools to make better decisions with one of the most difficult decisions that they face around surgery. So I think that's a really important point because, no matter how good, your healthcare technology is, it needs to be scalable, practical, and easy to adopt. And that's exactly how Cleo has been designed. So we're not just one test either. So we're actually bringing to market 3 tests that essentially cover the entire ovarian cancer landscape. So the first I've mentioned, that's the pre-surgical triage test. So think of that as symptomatic women. So that's essentially to help you identify whether it's benign or malignant. So this has the fastest route to market, so in terms of FDA clearance, because there's already a defined pathway and there's FDA approved tests out there. So that means that Cleo can do a 510K FDA approval, which gets us to the market the quickest. It also means that we can get closer to reimbursement earlier and commercial revenue. So I'll touch on those points shortly. The second test we'll bring to market is recurrence. So it's important, given ovarian cancer is really known to recur. Approximately 70 to 80% of patients, they experience a relapse after their first treatment, which is pretty shocking. So ultimately, the third one, and arguably the most important, is screening. So think asymptomatic women. So imagine a future where ovarian cancer could be detected through a routine blood test before any symptoms appear at all. So think similar to how we've got a pap smear for cervical cancer and a mammogram for breast cancer. So as we know, these two measures have been able to really dramatically increase the life expectancy of millions and millions of women. Whereas ovarian cancer, the landscape's actually remained really unchanged for decades. So the 5-year survival rate of ovarian cancer, it's 49%. So to put that into perspective, breast cancer, the 5-year survival rate is 92%. So there's a really alarming gap there that Cleo is trying to fix. So in summary, first triage, then recurrence, and then screening. So these markets collectively represent a multi-billion-dollar market opportunity. So each stage will build clinical evidence, it shows that we can execute, and it builds additional value along the way. So where are we now with our test? I need to slow down speaking, don't I? I'm rushing through it. Okay, so our test has already shown really incredible performance. So in previous studies, our performance has been 95% specificity and 95% sensitivity. Competitors or comparable tests on the market, they're operating at around 80%. So you can see there's a pretty large gap there. And I'll also highlight that for FDA approval, we're doing a substantial equivalence, which means that we only need to show that we're just as good or 1% better. So given that we're around 15% better at the moment, it's obviously very compelling. So now we're at the very pointy end of our clinical trial. So this will be our final clinical trial, which we call a pivotal clinical trial. So that's the equivalent of like a phase 3 in drug development. In diagnostic world, we call that the pivotal clinical trial. So once we finish this study, we essentially bring together all the data and we submit to the FDA, which we're hoping to do around Q1, Q2 next year. So very soon. So we've already ticked off one of the biggest milestones, which is patient recruitment. So we've collected over 800 blood samples prospectively across 19 US clinical trial sites, which has also helped to satisfy diversity requirements. I'd also want to point out that these samples are collected prospectively and not retrospectively. So to touch on that, prospective samples from the FDA are considered much higher quality clinical evidence because they demonstrate how the test performs in real patients in real time in the exact same commercial setting that they will be used when we launch. So from the FDA's perspective, this is considered basically the gold standard because it provides the strongest demonstration that our assay is working the way that it's intended to. By contrast, retrospective studies obviously work backwards to that. Researchers already know that the patient has got cancer and then they retrieve the blood samples and test that for analysis. So what's next? Ultimately, we need to test the blood that we've collected. So to do that, we need to go through a process called analytical validation. So in very simplistic forms, that means that we need to test our assay on 3 batches of manufactured test kits, just to show that our test is reproducing the exact same results and consistently doing so to satisfy the FDA. So that's the process we're going through at the moment. We started manufacturing with a company called Biotech. So they're a multi-billion-dollar NASDAQ listed company. So they're doing that at the moment. And once they've manufactured and sent the kits to our lab in Melbourne, we'll be able to start testing the blood. And that's a process that we refer to as clinical validation. So we'll test all the blood samples and we'll also have to test healthy individual samples and confounding condition samples just to make sure that the test performs across a real-world clinical population. And that's essentially it before we can submit to the FDA. So we're very much at the pointy end. It's been 15 years of R&D leading into this, but we've also been able to de-risk that along the way through really positive FDA meetings. We've had 2 of those in the past. So we're feeling really confident heading into this next period. But of course, what happens after launch? As we know, it doesn't really matter how good your technology is, but if it can't go to the people that need it and it can't be accessed by the public and more importantly, reimbursed, really what's the point? So Cleo's been really proactive in that instance as well. So we've established the US reimbursement pathway already, which I won't go into too much detail around, but obviously welcome questions. But essentially it means that we can start generating revenue the day we've got FDA approvals. So we initially will claim reimbursement through a miscellaneous code, 81599. And that will essentially allow us to start collecting real-world data and evidence that will allow us to progress through our dedicated CPT code over time. So what's the price of the test? It's probably a pretty big question that people would want to know. So the price of the reimbursement initially will be set through a process known as crosswalking. So they essentially crosswalk our tests to comparable existing biomarker tests that are already on the market. So today, those tests, which have shown quite inferior performance to us, they're getting reimbursed up to $900 per test. So pretty considerable when you imagine that in the context of what our total addressable market for this triage space. It's around 2 to 3 million women in the US alone. So if you extrapolate out those numbers, I'm sure you can realise that there's quite a fair few zeros attached to that revenue number, and that's just triage. Let alone when we get to the recurrence and the screening, which are multi-billion-dollar markets. All right, so to recap. So first, we've developed a simple blood test to address one of the most significant unmet needs today in women's health. Second, this isn't a technology we're starting from scratch. It's built around our patented biomarker, CXCL10, which is supported by more than 15 years of R&D, and a technology platform that has already demonstrated incredibly impressive, strong clinical performance, and is also recorded in 3 to 4 academic published papers as well. Third, the scale of the opportunity is undeniably significant. We began with a multi-million-dollar pre-surgical triage market, and then we expand into recurrence monitoring, and ultimately the much larger screening opportunity. Finally, we have a clear and very near-term pathway to commercialisation. We're now in the final stages of our pivotal clinical trial program, with FDA, as I said, on track for early next year. So for Cleo, the opportunity today is not simply to build another diagnostic test, it's to fundamentally change the landscape of ovarian cancer and change the lives of millions of women worldwide. Thank you.